Thank you so much to ARM for giving us the opportunity to presenting the business about CRC Oncology. Since the start in 4 years ago, the mission of CRC Oncology has been remained the same. Through our daily works, we're trying to bridging the oncology, the drug development, and eventually reshaping the landscape of cancer treatment. ARM has been no doubtful plays a significant role in promoting the growth of cell and gene therapy in the past decade. This slide cited the information posted on the ARM website, which gives a vivid picture about how blooming today is the cell and gene therapy area. From here, we can also see the bright future in the decades to come in this specific industrial sector. In the past 4 years, CRC has always been giving the priority and focus on the advanced cell therapy for oncology treatment. As you can see from the pie charts here, regarding the therapeutic area, 53% are really oncology assets. And for the therapeutic types, we have a 53% are advanced therapies, which are diverse across the different type of CAR T therapy, iPSC, TEL, exome, and oncolytics virus. So we have a successful regulatory submissions and active oncology trial management experience in all these areas. CRC is the full service regulatory and clinical development CRO. So we provide a full portfolio service as outlined here. So from the very beginning, for every sponsor we support, we do the IND gap analysis, and this really highly suggested to start as early as possible. Typically, we will review the submission standards, which the sponsor, and also identify the potential risk in regarding the already or available data, and also plan the study, and further advise the sponsor about the best strategy based on their asset. We certainly cover all type of regulatory submission. We have the advanced technology system to compare all the ECTD submission, and also do the publishing and the FDA gateway submission. In addition to that, we have a very strong regulatory medical writing team to really author all type of regulatory documents, including IND submission and all the meeting briefing package, CMC amendment, and also the later NDA submission. What I want to highlight is really regarding the regulatory strategy planning. Through the 4 years, we proactively identify the opportunity for different type of designation for sponsor, and we communicate this in a very timely manner and share the data needed to support such a request, and then successfully obtain the orphan drug designation, fast track designation in multiple assets for the different indications. And we will continue to support all the upcoming sponsors proactively on this perspective. So we do the full clinical trial management service. We have a strong clinical operational team who have the deep connection with the most prestigious oncology clinical sites. We also have the full DM service, data management service. We are long-term partner with metadata and the material. And we have the validated server, the production server, and also the test server to run the standard SAS programming for every trial we manage. So we also, on the behalf of our sponsors, to keep close communication with the regulatory agency, we take care of all type of meeting request submission, from the meeting request to the meeting briefing package, to the meeting minutes response. So as you can see here, the type of meeting really diverged from interact to even the component diagnosed meeting like a CDH meeting and certainly CMC meeting. So for all these type of meetings, we have the good experience in successfully manage all these regulatory agency communications. So here's the list about clinical sites. We have the active clinical trials ongoing, and you can see all these are prestigious comprehensive cancer sites. And in addition to the more than 10 full-time employee based in San Diego office, we also have around 50 hourly paid contractors across the country to run the clinical trial management and site monitoring. In addition to our fixed team, we also have multiple former FDA reviewers to support the regulatory advising in regarding CMC, non-clinical, and clinical. We certainly have the best seasonal industrial veteran and pros, also provide the advice and the consulting services for the different type of agency communication, especially for the end of a phase X meeting. So all these resources will support our current and upcoming sponsors significantly and to reach their business success faster. So from the very first day when we formed the CRC, because myself is an American Society of Quality certified quality editor, we committed to the high standard of quality management. So for each sponsor we engaged, we established a private, this sponsor-only website on the Microsoft SharePoint, which is a platform fully validated based on 21 CFR Part 11. So in this private website, we also have a company standard structure to cover the regulatory submission, CMC document, non-clinical document, and certainly the clinical trial TMF. So this website is open 24/7 to our sponsor team and our team. This private website is certainly an access control, version control, audit trail website. So our sponsor can really clearly see through click the information button on each library. They can clearly see what we have been done in the past 24 hours, which document we edited, who edited the document. Also, this system enabled fully review, approval, and sign-off procedures, and this all will be archived in record for potential future inspection or audit. We always use the most current ECTD template to file the regulatory documents. And we also have a multiple company standard template for the different regulatory documents to ensure the quality of the document. As you can see here, for every project from the very beginning, what we really want to establish is the spelling standard, like how we really spell the compound code or the asset code, how we really spell some commonly used specific technology term. And this spelling standard will be staying in the regulatory library, the root to, so every medical writers who are working on the program, they need to check the spelling standard and follow the standard from the very beginning. And also, we use the metadata, the tracker, to track the progress of all the regulatory documents. So through this platform and this system, we're just trying to maintain a very high quality, efficient, and productive platform to support the sponsor, save the money, save the time, so they can use more of their energies and effort on the real research works. So really talking about the, here I want to share some interesting observations from our past 4 years submission. And I'm going to go through 3 perspectives regarding the CMC, non-clinical, and clinical. So here is really the CMC. So what we want to share here, which is a very high level point, but are essential, is really about CMC. Is really about 60% attention should be given to the control when the 40% attention given to the development. The development is certainly important because development is the like a cornerstone to enable the production, enable the process is capable to generate the product. However, just to have a process is certainly just to have the process and the product are certainly not enough and even may not be sufficient to support a successful regulatory submission. Control plays more parts in this whole development process and the regulatory submission. So what we have been commonly asked and what we commonly observed are due to the process transfer from the research environment to the GMP environment, what document should I provide? How should I control this process? How should I make sure my transfer is successfully completed? And then how is the impact if I change the manufacture facility? And how is the impact if I change the manufacture process? So we have the most comprehensive and kind of in the cell therapy area is pretty in-depth experience in regarding all these questions. And based on case-by-case basis, we can always give you the most practical and also manageable advice so you can know what are critical things you must prepare and documented and presented in your regulatory submission. Talking about control, so it's really the material. Everybody knows starting material like cell banks, virus banks, plasmid, and also the critical in-process materials like the culture media. So those materials is the selection of the materials and compare, if you change the materials, compare the materials, and also archive all these materials, COA, and also the regulatory supported file. Those are very tedious but critical for the successful regulatory submission. In-process control, how you define your critical steps, how you really define what to test, the analytical method, the kind of reference range based on the specification, based on the acceptance criteria in the specification. Go to the specification. What we commonly see due to the advanced therapy area, which is very different from the targeted therapy or even biologics. Commonly we see the seamless process from the drug substance to drug product. So multiple questions are specific for this area. It's like, should I have a separate specifications for drug substance and drug product? Should I include the IPC in my specification, or should I just have one specification to cover the harvest and also the drug substance drug product? And also how to decide for some specific area, like for example, just for safety, for all this impurity testing, should I do it on the substance or on the product? So all these questions we have been commonly asked, and we have also successfully managed multiple submissions with the most comprehensive and practical specifications based on the development phase. Batch control, which is very important. What we commonly see is that in the beginning, especially for the research lab, people may not have direct connections between the research batches, even the non-clinical pharmacology study batches versus the tox batches versus the clinical batches. However, the batch information is really a reflection of your process development progress. Even in the IND stage, the process development whole history is not necessarily to be presented in the IND package, but certainly this batch control, especially each batch, what are the source material, what is the starting material you used, the purpose. So the batch control and also the justification of your specification around the batch control, around the process development, those are the most valuable experience and the most valuable supportive information for you to reach where you are when you submitted the IND. So from the very beginning, every time when we start to engage a new sponsor, when we talk to their CMC team, we always start from the batch information. So other than starting, so when we do the submission, typically we start like drafting the manufacturer, starting material, all this stuff. But when we communicate with our sponsor, always the very first point we start is up to now, up to today, how many batches, regardless the quality standard, even it's a research level batch, it's fine. How many batches you have been manufactured so far, and where, follow what specification, use what starting material. So only after we have a comprehensive tracking sheet of all these batches and use what analytical method to test the batches, we have a clear picture of the development history of this asset. So stability, this is a very tricky part, especially for the cell therapy. Everybody knows, especially for some autologous cell therapy, for the real clinical batch, it's almost infeasible to conduct the stability study on the clinical batch. But we have to do the stability study, otherwise we have no proof or no base to state the expiration date of our product. Then the question will be how to design the stability study, how to design the long-term, the accelerated stability study. Another thing has been commonly neglected is the injection in use or compatibility study under this stability study umbrella. So if it's to be injected through whatever, like a intracannular or IV injection or whatever, if we really planning to do the drug administration through any type of injection procedures, injection use stability study will be required in the IND package. And how to design this study appropriately is also very important. So multiple times, we are the one to alert our sponsors about the requirement of such a study, and we are the one to share our insights on how to design such a study and how to conduct such a study, and to make sure this has been successfully completed and included in the IND package. The other thing is really the shaping stability study, which is also quite unique and special for our area. So we also have a different, kind of depends on the product. We also have a different advice to how to design and implement or conduct such a study. Another thing we want to mention is really about this kind of a periodic facility inspection. Very, very different from all the other therapeutic area, other therapeutic types, like the targeted therapy, ADC, or monoclonal antibody. They have more time. They can really do a CMC development across 5 years or even longer from the IND to the NDA or BLA. And they can certainly switch the process maybe 2 or 3 times and switch the facility. But for cell therapy, even the, the gene therapy, we have to be super cautious because this is an area very highly likely one clinical trial will bring you to the registration. So from the very beginning, after you have some very preliminary process established in the like a research lab or the bench top, when you really transfer this process to your very first GMP CDMO, that's really the moment you need to start thinking about the potential BLA submission in next 3 years. So from the beginning, everything, the process, the control, material, specification, analytical method, and especially facility, we may need to proactively think and apply it as this will be the process and the place for potential registration batch, even commercial batch, especially for biotech company who are very, kind of like a fast-paced and more likely will hand over the asset to the big farm in the late stage of the development. This will be a critical point for the big farm when they're doing this due diligence through the BD process. So talk about the non-clinical. It's also very different from the, all the other therapeutic, types has been developed in the past. So typically for the non-clinical, we have the pharmacology, which is a potency study, and the PK, and also the toxicity study. For the cell therapy, it's kind of hard to really establish the most appropriate PK models, even kind of like a linear range or linear relationship associated with between the administration dose versus the in-body exposure versus the efficacy. So for the non-clinical, for the advanced cell therapy, it's really for the potency study, which is to prove the efficacy. It's a very much case by case. And also sometimes it's very hard to identify the, the most applicable or appropriate models. So CRC has been significantly support our sponsors multiple times in doing the literature search, identify the appropriate model, even connect the sponsor with the prestigious CRO who has the most advanced knowledge in the very innovative area in established model, find out the surrogate asset or target to prove the concept. Talk about the toxicity study. So the first thing, if it's not an oncology asset, say if it's iPSC cell for the cardiac disease, then this tumor genicity is really the top thing we must consider. And then go to the top general tox study, either the acute or chronic single dose or repeat dose. The study design should be more focused on identify the potential appropriate biologic effective dose and safe dose, and also the most appropriate administrative route. So for the cell and gene therapy non-clinical study, really 60% attention should be given to the toxicology study, and 40% attention should be given to the PD, the potency study. And certainly the PK sometimes will be hybrid into either the PD study or the tox study. But still, tox study is more critical in compared to all the other part of the non-clinical study. And also, yes, FDA has an interactive meeting guideline, which interactive meeting tends to be more focused on CMC and the clinical design. But still, interactive meeting is certainly a very good vehicle or very good opportunity if the sponsor has a doubt about their non-clinical study, especially non-clinical tox study design or commitment. This is really the best timing to proactively communicate to first state and positioning why the sponsor believe such a study is sufficient to address the potential concern on the off-target toxicity, the potential tumor genicity, or even the feasibility to prove the dose tested in the non-clinical can be transferred or converted into the human dose. So this is really the best timing to communicate as early as possible, so the sponsor will not spend the time, money, and effort later on, but still find out by the time when do the IND submission, some critical pieces are missing from the package. So for the clinical, design is a very, 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 very important. I can't emphasize the protocol design sufficiently. And really, I think the keywords of the design is also the control. It's kind of like a similar, like what I say in the CMC, it's really the control. The control of multiple things involved into the study. So the very first thing is really the target population. So it's the define. So understand the disease the best first, and make a very specific process definition on the target population. Have a very good control of the enrollment for this specific population. This set up the very solid base for the potential success down the road. And another thing is really how you really characterize the dosage versus the in-body in vivo exposure, and how you characterize, even investigate, even like analysis, the exposure versus the efficacy and the safety. So what we always do is if for CAR T therapy is kind of better than some other therapeutic types, because which multiple CAR T therapy approved, and which all this FDA information from the FDA, we already get some good sense about how to establish the evaluation about the exposure versus efficacy in this specific type of therapy. But when go to the other therapy, basically what we would highly recommend is the proactive communication with the specific KOLs in the therapeutic area. Because really, KOLs know the disease the best and know all the other ongoing treatment the most. More important, they deal with patients on a daily basis. They know how to evaluate the progress or the recovery of the patients. They have the best understanding about the most appropriate clinical evaluation in define the efficacy endpoint. So when go to the very, very innovative therapeutic area, what I heard multiple times, the agency FDA is highly encouraged sponsor take the leading role and actively interacted with the therapeutic area to come up with the creative but feasible and with a good scientific rationale endpoint for such a measurement and monitoring of the progress of the disease. So in this case, what we always do in the past 4 years is we always connect our sponsor with the top players in the relevant therapeutic area. And through multiple phone calls, we kind of come up with the critical, measurable efficacy endpoint to evaluate the progress of the disease after receiving the treatment. Other thing we need to keep in mind is really the visit and assessment, especially for the cell therapy and the viral therapy area, gene therapy area. The long-term safety follow-up has always been a commitment and a requirement from the regulatory agency. So when we design the protocol, we always consider 3 factors. Is what is the commitment of the acute toxicity monitoring? and what is the commitment of the long-term chronic toxicity monitoring, and what are feasible based on the the disease stage, and also the patient status, and also the affordability of the whole society. So when we put all these factors into consideration, the visit and assessment schedule in the protocol are really more realistic and practical. And also, this visit assessment schedule in the first-in-human protocol really gave the best information or set up the reference value for the future monitoring if the drug is approved. So operation is very critical. Operation compared to the other trials or other type of assets, more, I would say, tedious, comprehensive, require a lot of attention, require a lot of head count. So we certainly need to proactively planning this cold chain shipment and the inventory tracking. And also because for some specific asset, clinical site now is no longer just the patient to treat the patient. They're also involved into the manufacturing. They are also the place when the source material, where the source material is collected. So the site training becomes even more critical and important, and all the documents needs to be well prepared, clearly written, and well trained to make sure such collection or even the safety monitoring will be completed by the site staff with a good compliance. And certainly the site and the data monitoring. And so as I'm saying, similar like a CMC, because it's really highly likely, especially for the rare disease and for the very innovative therapeutic area, therapeutic type, your first in human trial could be your registration trial. Even in the beginning, that wasn't the plan in such a case. So periodic inspection on the TMF folder on the site, like a periodic GCP site inspection, is highly recommended. So talk about the CRC's service. We do fix the cost. We kind of all the IND, all the regulatory submission, even the clinical trial, the contract is company standard contract based on the milestone. And also it's like a unit priced clearly, no upfront payment. We do monthly bill based on the works completed in the past months. And this type of contract save a lot of time and energy between the sponsor and us. So both parties can really focus on the development works without, kind of a back and forth communication or negotiation on the continued change the budget or continuously change the cost or whatever. We understand the study may need protocol amendment, may need a change of the scope of the study. So for each protocol amendment, there's standard price, and it depends on the size of the amendment. It's also priced standard, like the small amendment, a big amendment. So we have the most transparency about our contract, and all our contract is very standard. We just trying to do this standardization to give our sponsor the best customer service experience, and also the most efficient communication. Save their time, save their money, and also save our energy for the real works. So we are very much, as I explained previously, we are certainly 100% US regulatory submission and clinical trial management full service CRO. We do provide consulting services to the China regulatory submission. In the early years, we also provide some consulting services, even like the medical writing support for the China IIT clinical trial, but we closed that part of business sooner after the first year. And currently, if any sponsor interested in learning more about how to really do the regulatory submission in China, we are happy to support with the consulting services, but we do not do the regulatory submission or clinical trial management in China. We are 100% US-based, US regulatory submission and clinical trial management. And this service is open to global sponsors, regardless you're from Europe, China, South America, North America, even in US, as long as you are the innovative drug developer. And actually here is like advanced cell therapy is our high priority, is highly welcomed. So we are happy to support you to be your US regulatory agent to support the development of your drug. So here's my contact information. If you have any questions, always feel free to send me an email. We are happy to support this entire industry sector. And so together we can bring more innovative, effective treatment to our cancer patients. Thank you so much.